I see people are starting to sign on. I'm gonna go ahead and share my screen as we get a couple more participants. Hello, hello. Welcome. Welcome to the webinar, Getting to Know Your Scientific Community. My name is Kirsty Bourne, and I will be your host. I have worked in applied research centers and labs, and I've been a corporate trainer, and I also worked as a STEM teacher for 7 years. And I taught over 3,000 students during that time, and I wrote a bunch of lesson plans, and my company wanted to do a webinar during when everyone's home, so we wanted to make sure that people still get to know some really cool scientists and get to think about science when they're learning from home. I'm just putting up a screen right now, you can see, and there's some information about the webinar. We have a Q&A section on the bottom of the screen, and you can click on that and ask questions at any time during the webinar. So even when the panelist is speaking, you can go ahead and type in your questions and it won't interrupt them. And I will go ahead and try to ask all those questions for our panelists when we have our questions time. And I wanted to note that when you ask a question, I'm not going to be saying your name. It's just because later this is going to be posted on YouTube, and just for security reasons, I won't be saying your name. But we love all of your questions, so please make sure to send those in as we're talking. I wanted to let you know that our webinar today is sponsored by my company, Elim Bio. Elim Bio is busy supporting researchers on the front lines. We're proud to say that we're helping scientists solve the problem of COVID-19. During this unusual time, we also wanted to reach out and help feed future scientific curiosity. We felt the best way to do this is give you at-home students a chance to meet some of the really cool scientists in our community. Today's panelist is Ryan Weiss. Ryan is an experienced educator and a scientific communicator, and he has a passion to inspire students to pursue careers in science. Ryan has his PhD in organic chemistry from University of California, San Diego. His current research focus includes using CRISPR-Cas9 gene editing to uncover what controls the production of sugar in our cells. And now I'm going to hand the floor over to who we've been wanting to speak to, Dr. Ryan Weiss.
All right, thanks so much, Kirsty. I'm going to get this shared. Should be able to see it okay. Is that correct?
Yeah, that looks wonderful.
Okay, great. Hello, everyone. My name is Ryan. I'm super excited to be with you today and share with you my research and tell you a little bit about genome editing and CRISPR, some stuff you might have been hearing about in the last couple of years that's really been changing the world, and it's really exciting stuff. And I'm really excited to be able to be a part of that and share with you some of my research. So the title of my talk today will be Genome Editing to Discover How Sugars Are Produced in Our Cells. I'm going to see if I can get this pointer on here. There we go, laser pointer. All right, so I wanted to first tell you a little bit about where I work. I work at the University of California in San Diego. So this is a big university. I work in the medical school, and specifically I work in this biomedical research building, and I work in a lab on the 4th floor here. We have a really big lab, so I'm just one person of many researchers in this lab, and we all work under a professor. His name is Jeff Esko, in the Department of Cellular and Molecular Medicine. So our whole lab group studies sugars, and I'll tell you a little bit more about what I mean by that. And my projects kind of come under this umbrella of what's known as glycobiology. So glycobiology is a area of biology that specifically studies carbohydrates. And I'm sure some of you have heard of carbohydrates. This is typically found in our food, right? So if you look at your ingredients on, for instance, certain sweets that we all love, like donuts or cookies, or in a lot of breads and everything, a lot of these have sugar in that, and sugar, carbohydrates, you can interchange using those different terms. So one of those sugars, I'm just showing you an example here, is called sucrose. So this is typically like the white sugar that you would put on your sugar cookies, for instance. And this is known as a molecule. And this molecule has different carbons and oxygens and hydrogens. These are atoms that are found in nature. And when you put them together, they form what's known as a molecule. And here we have two different molecules connected together. And this is two different sugar molecules that together form a molecule known as sucrose. And so these are sugars that we typically will eat and digest when we eat breads and our donuts and everything. And this is really important for our bodies to function normally and to give us energy. So our body is made up of not only different organs, but also different cells, right? So the very basic building block of our body is the cell. And the cells need sugars in order to function properly. So these are one type of sugar that we're probably a little bit more familiar with. But there's also another type of sugars that are made by our cells. These are called cell surface sugars. They're also known as glycans. This is kind of a scientific term that we use to describe these other types of sugars that, for instance, are put out on the surface of our cell. So here I'm showing you one cell. This is a bacteria cell. And you see all this fuzzy bit here. This is a picture of an actual cell. And all this fuzz on the outside, these are all sugars that coat the whole outside of our cells. And these sugars are typically, here I showed you sucrose, our sugar that you eat, is just two molecules joined together. But typically these sugars on the outside of our cell are multiple sugars all put together. These are known as polysaccharides, poly meaning many. So these are multiple sugars all connected together. We can also show them in this cartoon form sometimes. It's a lot easier to kind of understand, where each circle or square is a different sugar all joined together. And these are put out on the surface of cells. And they serve not only just to protect the cell, but I'm going to tell you a little bit more about what other things that they do. But what's interesting and important to realize, and actually it's not taught a lot in school, is that all of our cells are surrounded by these sugars, literally all of our cells. Everything from our brain cells to our skin cells to our cells that are found in our heart or in our livers. And here I'm showing a white blood cell. So this is a cell that's involved in our immune system that floats around in our bloodstream. So you can see this is an actual picture. It's called an electron micrograph picture. This allows us to actually take a picture of a cell as it exists in our body. And you can see the cell has a nucleus. It has a cytosol or a cytoplasm that you might have heard about. And then it has a plasma membrane that surrounds the cell. But then look at this fuzzy bit that's found just outside of it. These are all sugars that are found on the outside of our cells. And so this is just showing what type of cell, but there's many other cells where sugars are also found. Cancer cells also have these sugars on the outside. And also, for instance, if you look on the inside of our blood vessel, look at all these sugars that surround the inside of our blood vessel. Here I'm taking kind of a cross section. If you look at our blood vessel where our blood flows through, if you take a little chop of it and look on the inside, all this right here are sugars. So you can imagine when our blood is flowing through our blood vessels, it also comes into contact with these sugars, which are attached to these cells that surround the inside of our blood vessels. So just wanted to point out that these cell surface sugars are found everywhere, and they're very important for normal functioning of our body. This is a kind of a cool way I like to think about it. Like, what do these sugars actually look like on the cell surface? Well, what if we took an airplane and we flew over the top of this cell? You would see something that looks like what I like to call like the sugar forest. All these trees, right? This is a picture of a dense jungle, but you can see that the surface of our cell also looks very similar, where all the trees are different sugars that are just coating the outside of our cell. There's another way I like to think about it as well. What if you have like a tennis ball? You know, the tennis ball has all that fuzzy bit on the outside. All of those fuzzies are actually what we would call sugars on the outside of our cells. So just another way to kind of think about what these sugars actually look like on each one of our cells. It's pretty amazing, actually. So why are these sugars important? Why are they even there? So one thing is, if we could look back at this picture, it serves as a nice protective coating for our cells. So if anything wants to get into the cell or interact with our cells, it first has to come into contact with these sugars. So it's a nice protective coat. But also these sugars perform a lot of other functions. Here I'm kind of zooming out. This yellow bit is just one of our cells. And here we have a nucleus of the cell, which we'll talk more about in a little bit. But you can see it all on the surface of a cell. This is another way we like to show these sugars. All these sugar molecules are coating the surface of the cell. So not only is it involved in protecting the cell, but it's also involved in interacting with the environment of the cell. So all of our cells, whether they're floating through our bloodstream or sitting in our brains, they exist, as a cell unit, but they also have to interact with what's going on outside, right? And so what I'm showing here is that these sugars interact with other cells, for instance. They also interact with other molecules, some of those being hormones or antibodies. These are molecules involved in our immune system. And so these are all really nice and important interactions that our cells need to make with their environment. But what's also happens is that a lot of germs and pathogens take advantage of these sugars on the cell surface. So here I'm showing some bacteria. So these are germs that if you get sick, sometimes these bacteria get inside of your body, right? And what happens is that these can also interact with sugars on the surface of our own cells. And also viruses, which I'm sure you all have heard about the current coronavirus that is existing. The coronavirus has been also found to interact with these sugars. And this is really important for viruses because they first interact with these sugars, and then that allows them to get into our cells. So these sugars are very important for bacteria, viruses, and even different toxins that can come and interact with our cells. So not only are bacteria and viruses involved, but also cancer, which is a really sad disease that affects a lot of people. Cancer also uses sugars to help the cancer cell to grow. And it also is involved in cancer cells spreading through our bodies. It actually attaches to these sugars as it rolls through our bloodstream and gets to our other parts of our body. And this just shows again that different pathogens and germs interact with these sugars in order to get into our cells. And also you've probably heard of different diseases involved in our brain, like Alzheimer's, that has also been found that these sugars are very important for these diseases to take hold in our brains and form different plaques, for instance, these different ways that it can mess up our brain and cause memories to be gone. There's also many genetic diseases. And by genetic, I mean genes, and we're going to talk a little bit more about what that means later. But certain kids are born where they have mutations or mistakes in their genetic code where they cannot make these sugars properly. And that causes a lot of different problems that lead to what we call genetic diseases. And I'll talk a little bit more about that later. So because these sugars are so involved in not only many normal processes in the cell, but they're also involved in many diseases, there's a lot of interest in researchers trying to understand how these sugars are made, for instance, in cancer cells. And is there a way that we can control how these sugars are produced as a way to potentially treat these types of diseases, or for instance, to block viruses from getting into our cells? So again, we're going to return to this picture where we have the cell and it has all these sugars on the outside. But you might be wondering, well, how are these sugars made? Like, how do they get produced and then put on the outside of the cell? Well, it all starts in the nucleus of the cell. The nucleus you can think of as the brain of a cell, right? So the nucleus controls everything that happens in our cells. And each one of our cells has its own nucleus that, again, controls what happens. So my research is to try to understand how DNA in our nucleus can lead to changes in sugars on the surface of our cells. So what happens is that DNA, which I'll talk a little bit more about in detail if you forgot what DNA is kind of a blueprint or the instruction manual that tells the cell what proteins or what molecules it needs to make to survive. And one of those molecules are sugars. So what happens is that the DNA tells the cell to make certain proteins. And here I'm highlighting a certain type of protein called an enzyme. It's kind of almost another name for a protein. Enzymes are really important because they allow molecules to be made really quickly. And so enzymes are really involved in making sugars. So it all starts from DNA, which makes enzymes, which then makes our sugars. So we know how these sugars are made in the cell, but we don't really know what controls the production of these sugars. Why do cancer cells make a lot of sugars, for instance? So my research is specifically involved in looking at our DNA and how our DNA controls the production of sugars. Currently, we don't really know much about how this works, but we do know a lot about DNA. So I'm gonna switch gears a little bit, and we're gonna talk about how DNA is really important to make our cells function correctly. And at the end, I'm gonna return to kind of how my research ties in everything, including DNA in particular. So DNA stands for deoxyribonucleic acid. We just call it DNA for short. This is a kind of a cartoon picture of what DNA looks like. It exists as what's called a double helix. You got this side and this side. It forms what we call a double helix. It's kind of the spiral shape. So DNA is found in the nucleus of our cells. It's all packed in there. We have a lot of DNA. We have over 1 billion letters of DNA. And by letters, what I mean are these 4 different molecules that make up our DNA. We have C, G, A, and T. It's pretty amazing that what makes us who we are is all packed into our DNA. And what's really interesting is that each person on this webinar has very unique DNA, and it actually makes you who you are. And each of us has a unique DNA code. That's why I look, for instance, very different than Kirsty does, right? It's that we both have DNA, but we have changes in our DNA that make us distinctly who we are. And each of you has unique DNA that makes you even look different from your parents, which is pretty amazing, considering that a lot of our DNA we actually inherit from our parents. And we'll talk a little bit more about that in detail. But here I just want to introduce DNA, the molecule itself. It's shown here as like a cartoon, but keep in mind that DNA are these individual molecules that interact with each other to form this double helix structure that gets packed in to the nucleus of our cell. And it's the building blocks of life. How does DNA responsible for making us who we are? Well, I'm going to introduce you to, maybe some of you have heard about it. It's called the central dogma of biology. It's kind of this terminology that gets thrown around. But really what it means is that this is the building blocks of life, right? So DNA is this molecule made up of A, T, G, and C molecules. And what happens is that this DNA is an instruction manual to tell our cells what proteins need to be made. And this is a fairly complex process that I'm not going to go into in major detail, but really whatever our DNA says in terms of its A, T, G, and C molecules joined together, it tells the cell what proteins to make. And proteins make up what our cells and who we are. So it's kind of, you think about DNA as the blueprint or the instruction manual to tell the cell what proteins it needs to make. So here I'm kind of zooming out. We have our cell and then our nucleus. Our nucleus is packed with chromosomes. And really what a chromosome is just a bunch of DNA really wound really tightly together and packed in there. Because if we just let the DNA flow around, it wouldn't pack very well into our cells. But it's because our DNA is so long, it has to be compacted together into what are called chromosomes. So again, this is our cartoon picture of what DNA looks like. Different sections of DNA encode or give instructions for genes to be made. And I'm sure some of you might have heard of genes. And we're not talking about my blue jeans that I'm wearing, but we're talking about a gene with a G. So this is a DNA code that says you need to make this protein or this gene. We kind of can use that interchangeably. And here I'm showing what that means. So a certain set of DNA or a segment of DNA gets made into a protein. This is really important for our cells to exist. So genes are what kind of make up our DNA. And what's really cool about genes is that you inherit them from your parents. That's why we kind of look, that's why we typically look like our parents, right? Is that we are inheriting DNA, one copy from each of our parents. So here I'm kind of just showing that you have two different copies of a certain gene and you get one copy from each of your parents. That's why it's really important that you have, one copy from each. And I'm going to give you a little example about how important it is about what we inherit from our parents in terms of our DNA. So this is an example of a genetic disease. So typically we inherit a lot of genes that are really important, right? For our eye color, for instance. If both of your parents have brown eyes, most likely you're going to have brown eyes as well. This is determined by your parents' DNA, right? And if you have brown hair, sometimes most likely both of your parents will have brown hair. And it gets more complicated. Maybe one parent's hair is blonde, one is brown, and then you end up with blonde hair. And we're not going to go into that in detail today, but it's all dependent on your parents' DNA passed on to you and how it's passed on to you. So these are examples, right, of good genes that are passed on. Oh, okay, I have eyes. And not only do I have an eye, but I also have a color eye as well. So these are all good things. But sometimes what happens is that either randomly or passed through generations of your family, there could be a mistake in your DNA. And here I'm just showing, let's say we have these 3 letters in a row that in our DNA, G-A-G. Sometimes what happens is you can get a mistake where you have, instead of this A in the middle, you get a T. You might think, let's just say that is the only mistake in your whole DNA code. You go, everything looks great through your whole DNA code, but there's the one letter that's the wrong letter. And this was passed on maybe from your parents, for instance. What's crazy is that this one mistake in your DNA code can have really amazing, profound effects on your body. Here I'm showing one disease it's called sickle cell anemia. This is a disease where typically we have our red blood cells shown here. This is a picture of an actual red blood cell. It's nice and round, and our red blood cells are really important because they carry oxygen through our body, through our blood. But what happens is if you have this one wrong letter in this specific gene that encodes for a protein that helps you carry oxygen through your body, you have one wrong letter, just one. What happens is it causes our blood cells to look like this. It's really misshapen, and these cells are no longer able to carry oxygen through our blood. This causes major problems, if you can imagine, in someone who has this mutation and who has this disease. So this is really sad, right? Because sometimes you can't control it. If both of your parents have a mutation in this gene, there is a chance that you might inherit this mutation as well. So up until about 10 years ago, this was kind of a thing where you just had to deal with it, right? Unfortunately, you were going to get this disease, and it's really sad, right? Well, very recently, there's been some new technologies that have come out in science and in genetics. So genetics is the study of our genetic code, our DNA, that allows us to ask the question, what if we could fix this mistake in our DNA? What if there was a way to, instead of have a T here, we could change it back to this A molecule, and we could get a quote unquote normal gene back. If you reverted this back to normal, what would happen is these cells that have this kind of sickle, this like banana shape to them, would go back to being able to be normal cells, which would be pretty amazing, right? If we could do that. But the problem is, typically when you get your DNA code is what it is, and it's what makes you who you are, right? But what if you could go and change that DNA code? And this again was pretty impossible until recently, until a new area of research and a new tool called gene editing allows us to go in and actually change our genetic code in a very precise and directed manner. And this, so you can imagine this has amazing implications in terms of treating kids or any human with a certain genetic disease that involves a problem in the DNA code. So you might ask, well, okay, this sounds amazing, right? All right, we're able to change our DNA code, but what does that actually mean? How do we do that? What is gene editing? What does that even mean? So I'm going to try to help explain that a little bit. Again, we're going to go back to our DNA molecule here. So again, all of us have this DNA, and their DNA, I'm writing it kind of as if you would write it in a Word document on your computer. T, G, C, G, G, A, T, G. Maybe this first line right here, maybe this encodes or is an instruction manual to make one certain protein. And then this line tells to make a different protein. And what happens is in the nucleus of our cell goes through it and says, okay, we need to make this protein, then this one, then this one. But when we write it out on our computer screen, we just write it here as all these different letters. We need to remember that each of these letters is a different molecule, right? That the cell goes through and reads, just as if you're reading it on your screen right now. So gene editing says, well, what if we had this code in our Word document? What if we wanted to go through and delete a certain part? Or what if we wanted to add certain letters? Or what if we had the wrong letter and we wanted to replace it with a different letter? Gene editing is a tool that allows us to actually go into a cell and change the DNA in this way. It's pretty amazing. So today I'm going to tell you about a tool that has really revolutionized this field of gene editing and really what has defined what gene editing means today. And this is known as CRISPR. So CRISPR, it's a pretty fun word, but in reality, it's just an acronym that stands for Clustered Regularly Interspaced Short Palindromic Repeats. It's kind of a mouthful, right? That's a lot to say. And you don't need to remember that at all. But if you want to memorize that and impress your friends, that might be kind of cool. So you can go back through and maybe look at the recording of this, and you can look it up on Google and memorize what that means. But really all you need to remember is CRISPR, and it's kind of a cool, fun, easy to remember term. But what this really stands for is this is a gene editing tool, or the gene editor, as I'll call it. What CRISPR allows us to do is to edit DNA, not only of human DNA, which is amazing, but anything that has DNA, virtually all organisms. So we could edit DNA in, let's see, mushrooms, for instance. And I'll tell you more about that later. And you can edit the DNA of a mouse or a worm or an ant, anything that has DNA, because all organisms have DNA that is a instruction manual to tell the cells what proteins they need to make and what makes an organism what it is, right? So here, I'm going to tell you about CRISPR, because some of you may have heard about this. This is something that was discovered not that long ago, and it has revolutionized the world as we know it. And I'm going to give you some cool examples of how not only how it works, but how it can be applied. So how does CRISPR work? Again, we're going back to this model where this is a molecule of DNA, shown a little differently than before, but we have all our different letters. And let's say we have these two letters that are maybe the wrong letter. Maybe they were a mistake in this DNA, and we want to change it. CRISPR allows you now to go in and only change these two letters, not these ones over here, just these two. And let's just take a zoom out picture of what our DNA looks like. Our DNA is over 1 billion of these letters. 1 billion, that's a lot, right? And so how would I change just 2 letters in that billion letters? It's using CRISPR. It now allows us to go in and precisely change whatever letters we want. That's pretty amazing, right? To be able to go and zoom into a very, very, very specific spot in that whole sea of letters and change just that specific one. Well, where did CRISPR come from? In order to understand how it works and how we can use it, we first need to go back in time. And we need to figure out, well, where did this come from? Did someone just invent this in their lab? Well, actually, nature invented this. This CRISPR has existed way longer than we ever even knew it existed. And actually, one of the first places it was found is in our yogurt. So yogurt has bacteria, which I had talked a little bit about earlier. But in bacteria is where CRISPR was found. And we're going to kind of take a little, we're going to go through the timeline or the history of CRISPR a little bit. CRISPR exists naturally and exists as a defense mechanism or a defense system for bacteria. So here I show a little bacteria fighting with the viruses. Bacteria and viruses have been battling for eons. And just like we get infected with viruses, bacteria also get infected with viruses, and they have been for longer than we have existed. So here I'm showing, this is what a certain type of bacteria look like. We're talking about a bacteria called E. coli. That's just one type of bacteria. So this is another picture of a bunch of bacteria cells. Now bacteria, if you'll remember, these are germs that infect us sometimes, and they can cause different diseases and cause our bodies to have to fight them off with our immune systems, and it can cause different infections. Well, bacteria exist themselves as cells, and these bacteria cells can also be infected with viruses. And we'll talk about that in a second. Well, it was here that CRISPR was first discovered in bacteria. And this was back in 1987, which I'm sure is before most of you were born. It was when I was 1 year old. That's when it was discovered. And it was pretty amazing. All these scientists they found these sequences in the DNA of bacteria, and they were repeating sequences, and they were kind of out of place. And they're like, what are these sequences of DNA doing here? They don't encode for proteins that are needed for bacteria to survive. They seem kind of weird that they kind of repeat each other, and they must do something, but we have no idea what they do. So this was in 1987. Fast forward to 2007, a yogurt company found that these sequences actually encoded for a defense system against viruses. Again, this is another picture. All these little green guys, these are viruses, or they're also known as bacteriophages or phages. These phages can infect bacteria. It could be really bad for a yogurt company, for instance. So yogurt companies use bacteria, good bacteria, to make their yogurt. But sometimes they found that viruses would come in and infect and would kill off a lot of this bacteria. You can imagine this would be really bad for the yogurt companies, right? What they found when they studied their bacteria that were able to survive viruses that tried to infect them, they found that they were able to defend themselves against these viruses. And they found it was due to these repetitive sequences in their genome that allowed them to defend off viruses. So I'm going to show you a cool little video that shows you how this works. So here's our virus that comes and attaches to the surface of the bacteria. And what happens is, we're going to let it come back over again, that the viruses inject some of their viral DNA into the bacteria. That's what's happening right here. This is called viral DNA. And what happens is typically this will go in and allow the virus to reproduce in that bacteria cell, which is pretty much what a virus wants to do. It wants to reproduce as much as possible. So just like the coronavirus right now, it's not actually alive technically. It is a piece of RNA that wants to just reproduce, and it can't do it by itself. It needs something to do that. So in this case, it uses bacteria to do that. So once it goes in, typically it would be able to insert into the bacteria genome and get made, and then more viruses would get produced. But what they found was that once a bacteria got infected with this viral DNA, what it would do is it would come and put that viral DNA into its own genome. Isn't that amazing? It comes and be like, okay, we're going to remember this virus. So it puts that viral DNA into its own genome. So next time this happens, it remembered this virus based on the viral DNA that was injected in. And it has these proteins called Cas proteins that are nucleases that cut DNA. But the problem is these proteins don't know where to cut. They just know how to cut DNA. Well, what happened is because they saved this virus DNA in their genome, it was able to tell the Cas9, or the Cas proteins to go and cut up the viral DNA when it came in to infect. So this is how it defends itself against viruses. Pretty amazing, right? It's like a memory system. It's like, oh wait, I've seen you before. Hey, let's go cut up this viral DNA so it can't do anything to us. So again, this CRISPR system, as it's called, has been around for a long time, and it first exists in bacteria. So we're going to talk a little bit more about, okay, how does it cut DNA? What does that even mean, right? So again, remember, it saves that viral sequences of DNA in its bacterial genome and said, okay, next time it comes, we're going to remember it. So how does this system work? And this is going to translate to how we're using it now in human cells as well. So typically you also see CRISPR will also be called CRISPR-Cas9. And what the Cas9 means just stands for this protein that's able to cut DNA. It's called a nuclease, but you don't have to remember that, but just know that this Cas9, I like to remember it as this molecular scissors. So Cas9 is able to come and cut DNA. But the problem is that, like I mentioned before, it doesn't really know where to cut. It has to be guided there and needs someone or something to tell it what DNA to cut. That's where this guide RNA comes in. So RNA is a very similar molecule to DNA. I'm not going to go into major detail, but it's also made up of a bunch of letters. This guide RNA actually matched the viral DNA that was put into the cell. That's how it was able to tell the Cas9 to go and cut where it is. So again, CRISPR is basically two components. One is the guide RNA. These are the guys that say, hey, we'll tell you where to go cut DNA. We need to go cut this viral DNA up. And then over here, you have your molecular scissors, which is the Cas protein or the Cas9 protein. So it's just these two components that are able to go and cut DNA. And what's amazing with this system is it's able to go and cut precisely at a specific spot. So what happens is both of these guys join up together. You have your scissors and you have your guide, and they form what's called a complex. This complex will then go and cut DNA. So here we have our DNA, and this is a video just showing how this complex goes and can actually cut DNA at a very precise location. It's pretty amazing. So what happens is the guide RNA tells the Cas9 where to go and cut. And I wanted to show you this next video because this was revealed in 2017. This is an actual video of the CRISPR system in the cell cutting DNA. This is actually a video of this happening. Pretty amazing. So here we got this yellow kind of fuzzy bit. This is DNA. This is actual video of DNA. And this yellow guy is the Cas9 and guide RNA complex together. And if you watch, it binds to DNA, and here on the right, you'll see it actually breaks up the DNA right here in a second. Boom, DNA falls away. Pretty amazing. This blew scientists' minds when they saw this, because up until 2017, which was only a couple of years ago, this was all based on not seeing it actually happen on video. It was all predicted based on experiments that scientists had done. So this was the first time where we really saw it actually in action. So again, this CRISPR system allows you to specifically target DNA and cut DNA. Well, what is that? That's cool that bacteria are able to do this, right? Well, how does that apply to me and you as human beings? Well, in 2013, some really smart scientists said, well, what if we could use this CRISPR system and put it into human cells? And what if we could cut DNA precisely at certain spots? And so that's exactly what they did. And here I'm just showing a little bit, this is a little more detailed picture, but we have our red Cas9 protein, and it needs a guide RNA, which is this yellow bit. This guide RNA tells it where to cut in the DNA. So what these scientists found was that you could put any DNA sequence here and say, I want to cut at the gene that's involved in sickle cell anemia, like I mentioned earlier. I want it to cut right there. And what happens is when you cut DNA, the cell tries to, this is now in a human cell. We could cut DNA with CRISPR and the cell will try to repair it. Like, oh no, DNA is cut, we need to put it back together. Sometimes it doesn't do a good job of that. So what happens is you can actually introduce mutations at certain spots. So this would typically be bad, right? You don't want mutations. But what if you are a scientist like me and you want to know, well, what happens if a mutation exists at this spot or this spot? You can now do that in a very precise way using CRISPR. What's even cooler is not only can you introduce mutations at specific spots in the genome, but you can also put in new letters at a very precise location using CRISPR. And this has a lot of really cool implications that I'll talk about a little bit. And one of the first uses of this was in 2015. And they did this in mice because we can't do it in humans yet because we're still testing it. What they were able to do is they were able to cure a mouse that had a genetic disease by using CRISPR. So the mouse had a mutation in its DNA. And then they were able to send in the CRISPR system and cut right there at that specific mutation spot and put in the correct sequence. And what happened is they were able to cure that mouse. Pretty amazing, right? So what happened after these discoveries is CRISPR went everywhere. It blew up in the scientific world because it really opened up the possibilities of what can we do to, for instance, solve genetic diseases using CRISPR. Can we change DNA at the functional level in human beings? Or what about, can scientists use CRISPR to understand how our DNA works? And so this really exploded. And here I'm just showing some really cool examples where, for instance, they were able to make mutations in wheat where they were able to be resistant to mildew. So mildew is a big problem in crops. What if you could make wheat resistant to that? So they could use CRISPR, cut the DNA, and make them resistant to mildew. Here, they made mushrooms that don't even brown. This is just by making a mutation in DNA. They've also been able to alter human cells, and for instance, our immune cells, to attack cancer. So you can change the DNA of specific cells in our body so they can go and then kill cancer cells. Or what if we could prevent mosquitoes from carrying the malaria parasite, which is a huge problem in Africa and other parts of the world? You can now go into the genome or the DNA of mosquitoes and prevent them from carrying malaria. Pretty amazing. In terms of human beings, what if you could go in, let's say two parents know that they might pass on a mutation in their DNA to their kids. What if you could go in and change the embryo or change that little baby, the DNA, so they don't have, for instance, sickle cell anemia or muscular dystrophy or Huntington's disease? This is an actual possibility now with CRISPR. It's pretty amazing. Currently, actually, they're using CRISPR in human beings to solve childhood or to cure blindness in kids. They take the CRISPR molecules and they put it into the eye. And what happens is it goes into the back of your eye and it corrects a mutation in your certain cells in your eye. This allows people to be able to see again. It's truly amazing technology that has really opened up the doors to allow us to ask all these questions and see if we can solve diseases. So that's just some really cool applications to CRISPR. And I'm using CRISPR in my own research to try to understand what if I put mutations in certain genes in the DNA of our cells, how does this affect these sugars that are found on the surface of our cells? And these sugars are involved in all these different genetic diseases. And what if you could change these sugars to prevent viruses like coronavirus to get into our cells by using CRISPR on the DNA level? So it's been really amazing to learn not only how to use this tool. I forgot to mention, what's really cool is this tool is so easy to use and it's so cheap. It's about $50 and you can do an experiment and you can alter the genome of a human cell. All I have to do is say, what gene do I want to change? I just look for that DNA sequence, and I send in the two pieces of the CRISPR system, this protein that cuts DNA, and I tell it where to cut with a guide RNA, and it will go and cut. And it's so easy to use, and it's so precise in terms that it will go through our whole billions of letters, and it will go to a direct site and be able to change it. Pretty amazing. Hopefully I was able to explain it. It's very complex, and I know there's probably going to be questions, but hopefully thank you for listening, and just wanted to kind of introduce you to how CRISPR works. And really it comes down to just those two pieces that come together and allows us to alter the genome. And obviously there's a lot of implications to that. I didn't have time to talk about today, but there's a lot of really cool stuff online and really cool videos on YouTube that I can share with you that can share more about what we're doing with CRISPR. And so thank you all for listening, and any questions I'll gladly take.
Wonderful. I have some questions for you, some really fun ones.
Yes.
All right. The first one is, some superheroes have mutations, and I think they heard you talk about mutations. They were wondering, if superheroes had kids, could their kids have the same mutations? And can you make a superhero?
Yes, those are awesome questions. And the answer to both is yes. So some mutations are good, right? And that's what the question, whoever asked the question is kind of alluding to. And that's great, is that not all the time are mutations bad. Sometimes a mutation will allow a protein to work better than it did before. And this is definitely something scientists like myself are using. And a lot of treatments for different diseases, for instance, use proteins that work better than they did before. Or, like you said in your question, what you can do is you can introduce a mutation that actually makes a kid even stronger, for instance. What if you introduce a mutation in a gene in someone's muscles that actually made the muscles stronger? That's definitely possible, and they've done that in mice already. That's typically what we use in the lab, because we can't do all our experiments on humans right away. We have to experiment with animals that are very similar to humans, and then we kind of take it from there. So yes, if two superhumans came together and had a baby, most likely they would also have the mutation that makes them strong or really fast or really tall, whatever the superhero had. And so what about me? What if I would have wanted to become a superhero? That's possible too with CRISPR as well. The thing that that we scientists have to take into our thought when we're doing these types of experiments, for instance, on mice, is if we change the DNA of this mouse in this way that we think will make him into a super mouse, could it have any effects that would be bad? So you could be changing one thing that maybe makes the mouse really strong, but what if the mouse then is blind or can't see anymore, or no longer can really think really clearly, or doesn't know how to eat anymore? So we always have to take that into account, as if you change one thing, how will that impact other parts of the mouse? Or maybe, for instance, being smart, it's not due to one gene in our DNA. It's due to many different things. So sometimes to make a superhero, you would have to change multiple genes or multiple parts and make multiple mutations. So hopefully that answers that question. Great question.
That was fun. It was a good question, wasn't it?
Yeah, that was great.
Another question that came across was, they said that sometimes when they are reading, they misread a word. And they're thinking about your analogy, does a cell ever misread DNA?
Yes, no, that's a great question. And yes, there's a whole area of research looking at ways that sometimes the cell will accidentally skip over certain letters or will not read correctly how it's supposed to. So there's a protein in our nucleus where all the DNA is stored that goes and reads along the cell. And it's very complex, how it actually works, where it says, oh, you need to read here, but not here, read here, read there. And so sometimes what happens is that you can misread certain letters, or the cell will have a mutation that will cause the cell not to read in the correct place. So that's definitely a possibility. But typically what happens is if you have your DNA blueprint, your cell will read that blueprint. So that's why CRISPR works so well, is that if we change the instruction manual, your cells will typically turn out that change that happens. So it can get more complex where you have different ways that our DNA is read that can be changed. But yeah, no, that's a great question though, in terms of proofreading and can our cells correctly read? Yeah, and that's, you know, when you're talking about cancer, sometimes that's what comes into play, is that sometimes our cells read too much, for instance, read more than they're supposed to. So they make the DNA, they take too many of the instructions or too little. So that's a good question.
Wonderful. I wanted to move on to some of the questions that we like to ask our panelists on the regular basis.
Yeah.
Do you have people you collaborate with when you do research, or do you sit at your lab station by yourself all the time?
Yeah, no, that's a great question. So as a scientist, I always am collaborating with people. I typically am not sitting at my bench by myself. I work with a lot of different people. Even within my own lab group, we have people that are experts in all different things, whether experts in bacteria or in cancer or in working with mice. So my expertise is more looking at DNA and working with human cells, but I collaborate with people that help me when I need to learn about all these other types of fields of research. And another thing you imagine we have so many letters in our DNA, right? And how do you look through all of that? And how do you understand what the DNA means? So I work with another lab that they're experts in using computers to look at DNA. And so these are called, the term for them are bioinformaticians. It's a very cool word, but what they do is they use computers to understand our DNA. I am not very good with computers. I know how to use Microsoft Word and certain programs, the internet, but I don't know how to use computer programming as well. So I need to collaborate with people and work with people that know how to do that. So that's one of the main people that I work with that help me look at DNA, for instance.
Yeah, I love that. I love how in science we collaborate and are appreciative of everybody's different skills.
Yes, exactly. It's a huge part of science is working together. And all the people that I talked about that discovered CRISPR, they were not working by themselves. They worked together with a lot of people.
Wonderful. And do you just do science all the time, or do you have other things that you do besides science?
So I love science. It's really fun, but I definitely love doing a lot of other things in life. And I always think that it's really important, no matter what you end up doing, that you have a good balance in life and you do things that you love to do. And also, hopefully you love what your work is as well. And I'm really blessed to be able to really enjoy the work I do, but I'm not in the lab all the time. I love playing music. That's one thing. I play guitar and my brother plays drums and we have bands that we play together. So I play a lot of music and record music and play music different places in San Diego. And so that's something I really love doing. And I also have a family. I just had a baby girl, Zoe, that I love hanging out with, and my wife, Christina. And so I love hanging out with friends and with my family and playing music and just really enjoying life. Because I think when you can enjoy life outside of work, it really helps you enjoy your work as well.
Yeah, definitely. I agree. That's really cool. I love that you play guitar. How awesome.
Yeah, it's really fun.
How did you become interested in science? Was it something you knew you wanted to do, or did you have a path to get there?
Yeah, that's a great question. I always was interested in science, just in elementary school and middle school, but I think my interest really grew my freshman year, my first year of high school. I had an amazing biology teacher. This teacher, we called her Mrs. V. She also taught chemistry and all my other science classes at my school. And she was just an amazing teacher and really inspired me and encouraged me to pursue science, and just really showed me that learning science can be really fun. We did all these labs where we were dissecting animals or doing chemistry and different experiments. And that was the first moment I was like, you know what, I really wanted to do this. And I didn't know if I wanted to maybe go the medicine route, become a doctor, or do scientific research. So it wasn't until I went to college, and every step along my path, I've kind of knew I wanted to learn science, but I didn't know what science I wanted to do. And I kind of just took every opportunity that was kind of thrown my way. And I said, well, I'm gonna go try that and see if I like it. And I initially really loved chemistry. I still love chemistry a lot. And that's kind of what I got started in science. And I loved learning about how molecules interact and how we can make molecules, how those can be turned into treatments for diseases. And then as I learned chemistry, I also then was able to take my chemistry and apply it to human cells, for instance, to biology. And that's where I got interested in biology. And I learned CRISPR 5 years ago. I never even knew it existed until then. So every step along my path has been really opening my eyes to new areas of science and really asking like, well, I'm going to go learn that too. Or maybe I can ask someone to help me learn that. And it's really been really fun path to do that.
I love that. I know a lot of times I think kids think like whatever they're going to study is what you're doing, but sometimes your path kind of winds and
Oh, yes.
where different than you initially intended on it ending.
Yes, 100%. That's been my path. It's been one of these, yes, which makes it more fun, right? So,
Yes. More exciting for sure.
Yes.
Did you have any changes to your lab or what your working look like with everything with COVID-19 or?
Yes. So I've been working from home. So a lot of what I've been doing now. So typically when we work in a lab, I have my projects that I work on, and I get help from my boss, which is a professor, and people around me. So we do a lot of actual experiments in the lab. But then after you're done with your experiments, you then get to write up what we found, and we get to send that out to the world. And so we get what's called published. So we get to put it on the internet and in journals and magazines. And so what I've been able to do now is mainly focus on writing up my experiment results, and so I can hopefully share them with the world. So I've been working from home mostly on the computer, but I also have people that I collaborate with, that I work with, that are able to go into the lab. So our lab is starting to open up a little bit, but it was shut down for a while because we didn't want people to be close together with the coronavirus. So we all wore masks, and there was only a couple people allowed in there. And that's still how it is in the lab currently. So I've been staying home mostly and taking care of my baby and working on the computer. But I'll be going back in the lab here and there in the next couple of weeks, most likely. So, yeah.
So the students that are interested in science, it sounds like, should still pay attention in language arts class.
Yes, exactly. It all comes together. Yeah, writing is a huge part of science as well. And, initially I didn't like writing as much, but I think it's one of those things, the more you practice, and typically you have help from others that help you do the experiments. They also help you write as well. And you really learn the more you do it. And it's really fun to share with people, because you can't just call everybody up or, and tell them what you did and the exciting results that you got. There's got to be other ways, and one way is writing. So it's really fun to be able to learn how to communicate science. And communicating is not only writing, it's doing what we're doing now and talking about it. And so I get also a lot of opportunities to share my research like I'm doing now, whether it's over online or in front of people, and I get to talk about it. And when you talk about science, like what we're doing now, it really helps you learn too. I learn just as much telling other people about my science as you do listening. So whenever you can, if you're learning new things, whether it's in science or history, other thing, talk about it with your friends. And I think that's when you really can really learn a lot about not only how to communicate, but what the subject is that you're talking about.
Yeah, and I find I speak differently depending on what my group is that I'm talking to.
Yes.
And it helps me understand what I'm talking about better, because I have to think about it from different angles.
Oh, I think that's huge. If you just talk to everyone that knows the same thing that you don't learn much, right? It's really when you talk to your friends. And I talk to my friends just like I just did with you guys. I've told my friends what CRISPR is too, and just because it's really exciting, and a lot of people don't know what it means, and they see it around, you know, and hear about it. So it's really fun to be able to talk about it with people.
That's cool. Well, we're coming near the end of our call, and I wanted to thank you so much, Ryan, for being on our call. We really appreciate your time and your expertise, and telling us about your journey to become a, you know, expert scientist and
Still a lot to learn.
Oh, all of us always have a lot to learn.
Yes, no, you keep learning your whole life for sure. And yeah, thank you so much for having me. I really appreciate it. It's been a pleasure.
It was wonderful, and have a great weekend.
Thank you. You too.
Have a wonderful summer. And just remember, our friends that are on the call, to always stay curious.
Yes, stay curious, and if you ever have any questions, you know, keep asking away.
Wonderful. Bye-bye. Thank you. Bye,
Bye,
friends.
guys.
Thanks.
Bye, guys.